So in this chapter, we're going to focus on how we can control microorganisms in our environment as opposed to within the body, which would be antibiotics. So of course, in healthcare, it's important to communicate properly what level of control has been used or is needed in any particular situation. So when we want to control them outside the body, we have some terminology we're going to see. So when we use the word disinfection, this is either the removal or destruction of all vegetative or living, actively reproducing pathogens, but not necessarily endospores. So this is usually going to be the term we use for a reduction in the number of organisms on inanimate objects, as opposed to sterilization, which is the word we would use to describe the complete destruction or removal of all viable organisms. And again, sterilization is a word we would restrict to using on inanimate objects. If it's human tissue, we use the word axenic because that means there's no resident microbes. But we really can't sterilize the skin, for example, because we have hair follicles with bacteria living deep in those follicles. So it's kind of a misnomer when we say that they're going to do a sterile scrub before surgery, but it's the best we can do. So the terminology sterilization generally refers to the removal of all organisms. We now know, of course, prions are difficult, so this definition does not necessarily include prions. If we say antisepsis, we're talking about removing organisms on tissue or body surfaces, right? And so we would use antisepsis. Sometimes they call this degermation or dederming. Decontamination or sanitization would be physically removing or mechanically scrubbing those organisms off of the surface. Again, decontamination or sanitization we would refer to for inanimate objects typically. Now don't confuse the second word with sanitation, right? Sanitation is the management of refuse or garbage, not the same thing. That one syllable changes the complete context of that word. So you'll also see when we refer to different modes of control, either the suffix sidal or static. And so if it ends in sidal, it is something that can actually kill the organism. But of course, this requires proper use. If it's not left on long enough, it may not fully kill everything. So of course, any of these methods we're talking about, it's got to be used properly. So if it's bactericidal, it's not necessarily killing endospore stage bacteria, just the vegetative or the living reproducing stages. If it's fungicidal, it can actually kill any of those life stages. So fungal spores, hyphae, and yeast can all be killed with a fungicide. Virucides, again, these aren't necessarily living organisms, so we would just say that it inactivates viruses. And typically these are going to be used on living tissue. Sporicides, it's kind of a misnomer because when we say spore, we mean fungal spore, but a sporicide is usually targeting those bacterial endospores that are really hard to destroy. If something is a germicide or a microbiocide, this is really just a very generic term. It's kind of like saying antimicrobial. It doesn't really specify which kind of organism it kills. So we'll talk about some of these that are more specific and some generic terms that you might hear. Now those agents that can only inhibit the growth would end in static. So if it's bacteriostatic, it can at least prevent the growth of bacteria. If it's fungistatic, it can inhibit the growth of fungi. If it's an antiseptic, it may only go as far as being static, as in only inhibiting microorganisms. And typically this is the case because if it was cidal, it might be too toxic to use in or on the human body. So when we get to talking about antibiotics, on the other hand, antibiotics, if they were only static, would not be very functional because you don't want to just slow down the growth in the human body. You actually want to kill those organisms. And so we have to approach that very differently than we're going to do today when you're dealing with surfaces or equipment or things that can tolerate these toxic substances potentially. So some other terms we're going to see along the way: sepsis. So everybody's probably heard this term. This is basically what we use to describe a bacterial infection in the blood or other tissues in the body, which is why the word asepsis is referring to preventing those organisms or those infections. And so typically asepsis, this word can be used in many ways. You can refer to it as a practice or a field, right? We might have an aseptic field. But when we use aseptic techniques in the lab, we're basically doing those steps to basically keep the organisms where they're supposed to be and preventing contamination or infection of the tissues. So a wide range of results can happen when we do aseptic techniques. Some of them are going to be, good enough to call it sterilization, but most of the time it's just going to be antiseptic or disinfection on the other end of the spectrum. So again, the difference between antisepsis and disinfection is really what is being treated. So when we call it an antiseptic, we're really talking about those body tissues or body surfaces, such as skin or mucous membranes. If you have an open wound or a surgical incision, that would require antisepsis. So this is what we're doing when we prepare the skin before surgery or before an injection. We might use alcohol. Typically we use betadine and other iodine-containing compounds to prepare surgical sites. If you have an open wound from dental surgery, like a root canal, they might use hydrogen peroxide. But keep in mind, hydrogen peroxide is very toxic to living cells. It'll actually burst those living cells. So it's not something you want to use long term on a wound because it'll actually slow wound healing. But initially it can kill any bacteria that are on the surface of that open wound. So it is typically used initially on maybe an injury site or a surgical wound. When we do hand washing, running water over your fingers after using the restroom is not technically enough to remove microorganisms. But when you do hand washing, which is a physical process of degerming those tissues, along with a soap that can actually kill anything it comes in contact with, then you have a good example of Now, how we choose the method that we're going to use in any particular circumstance depends on what needs to be done, what's being treated, and things like that. But ideally, in the healthcare system especially, we need these methods to be inexpensive, right? If you're a healthcare system, you're probably using thousands of gallons of antiseptics and things over the course of a year. You need them to be fast acting because you want to be able to use that patient room efficiently, right? If you're going to have some method of control that you have to use that needs 10 hours between one contact and the other, it's probably not going to be very efficient. So it needs to be fast acting, non-toxic. It needs to be stable so that when you order, gallons and gallons of worth of a compound, that it doesn't become unstable or toxic while it's in storage. But most importantly, you want it to be selectively toxic, right? Obviously, you don't want to use something on a surface that a human body might come in contact with that might be toxic. So again, we want to target them without hurting us. And that also is something to consider when you're talking about surfaces that people touch. If you are trying to sterilize something, sometimes it depends on what you're working with, if it can tolerate that substance or that process, right? So solids like metals are easily sterilized using physical methods like heat or things like that, but it may not be suitable to other methods that might cause it to rust, right? If it's moist heat, that's a problem. Sensitive liquids or powders, right? If you're trying to filter an anti-sera that's going to be injected into a human being, clearly it needs to be sterilized and not just disinfected. Powders obviously can't tolerate moisture, so that would not be the best way to sterilize those. So it really depends on the circumstance. But there's really no universal method that works well for everything. When we decide which method to pick, of course, again, it depends on what's being treated. So obviously human tissues can't tolerate harsh chemicals or extreme heat. What the equipment is going to be used for is also going to have an impact, right? If you're going to be doing surgery and bypassing the protection of the skin and mucous membranes, you need it to be sterile. If it's going to be an invasive procedure, you absolutely need it to be sterile. However, if it's something that's only going to be used superficially and just touch the skin, of course, skin is very protective. It's our first line of defense. Then that piece of equipment could just be disinfected. Now, there's some controversy over things like catheters, right? Now, if it's a urinary catheter, it's sort of a gray area. Historically, people used catheters repeatedly at home, and they just simply disinfected between each use. Clearly, you wouldn't want to share a urinary catheter because it is introducing microorganism potentially deep into the urethra. If it's a venous catheter, however, it's going into the bloodstream. Clearly, that needs to be sterilized. Of course, in today's day and age, everything is disposable. It's all plastic, and so there's no worries about reusing some of these things. But historically, people did. So when we're talking about a catheter for, again, a urinary catheter, as long as the mucous membranes of that urethra are intact, it's not technically an invasive process. So disinfection should be enough if it's properly done. In the medical field, you may have those equipment or devices categorized into one of these three things. So if it's going to be coming in contact with tissues that should be azenic, meaning there shouldn't be any bacteria there or there are no bacteria present, of course, that's going to require sterilization. So we would call those critical medical devices. Semi-critical would be those that come in contact with mucous membranes. Obviously, mucous membranes are less protective than skin, right, because of the keratin. So mucous membranes have mucus, but they're also living, moist tissues that are more easily damaged or ruptured. So this would require a high-level form of disinfection. Sterilization would be preferred. So again, when we talked about those catheters, that would be the preferred method. But because that mucous membrane is supposed to be protective, there's a little bit less critical nature here. Non-critical devices would be those that only touch intact skin. So again, a stethoscope, for example. And so we would use what we call low-level disinfection methods in that case. Now, unless those pieces of equipment came in contact with blood or other bodily fluids that were potentially contaminated, then that would be something that requires a little bit higher level disinfection. So no matter which of the method we talk about here, you're generally going to have two modes of action. So if you're an antimicrobial agent of any kind, you're usually going to target one of two things. You're either going to try to alter or damage the protective covering of the microorganism that you're targeting. So for example, if it's an organism that has a cell wall, that's a really good target. Why? Because it protects the cell from osmosis. So if you can damage the cell wall, osmosis is really the thing that kills that cell. If you target a membrane, obviously the cytoplasm will no longer be retained or contained, but also the control that cell has over what comes and goes will be damaged. Of course, cell membranes are harder to target because they are similar across most living cells. Viruses, we have to target them differently as well because their coverings are different. So recall that viruses sometimes have an envelope, which is a phospholipid bilayer. But why we can target that effectively is because, recall, that's where they have those host recognition sites. So if you can damage the envelope, you take away the attachment site, you take away the ability of that virus to replicate. If it's a virus that doesn't have an envelope, what we call naked or non-enveloped viruses, then we would have to target the outer covering, which is the capsid. And these are actually harder to kill. You might think an enveloped virus might be better protected because it has a capsid and an envelope. But if you're a naked virus, the only covering you have is a capsid, which means they're usually much thicker, much sturdier, which means they're actually harder to kill. The other way we can target living things would be to go after their functional molecules, right? Their proteins or their nucleic acids. Remember that proteins are easy to denature with heat or acids, but if you unfold those functional proteins, if they lose their three-dimensional shape, maybe you've shut down enzyme activity, maybe you've disrupted receptor sites on the surface of the cell. If we want to go after proteins, we can either denature them or we can stop the production of those proteins, right? If you're a metabolizing organism and you're trying to replicate, you need to make proteins. So if we can go after those ribosomes, and recall we have the two different types of ribosomes, if we can go after 70S ribosomes of prokaryotes, then that's a good target. If we want to go after nucleic acids, this can be a little bit more difficult in the sense that all living things also have nucleic acids, DNA and RNA. So if you can manage to target the nucleic acids that are unique to that organism, or maybe the enzymes that are unique to that organism. So for example, retroviruses, if you recall, we have reverse transcriptase enzymes. That is one of the targets when we get to antibiotics that we talk about. Antiviral medications do go after those enzymes. So if you can either destroy the nucleic acids directly, or you can cause them to replicate inappropriately. So we're going to talk about some antiviral medications that basically cause the virus to replicate wrong and cause mutations in their sequence, which makes them non-functional. They're going to have basically try to move on to the next generation with junk DNA, and so they're not going to be able to do anything. So when you look at a chart like this, what we talk about the relative susceptibility, recall that we talked about how endospores are really the thing we target when we autoclave things in the healthcare system. And it's because we understand if you can kill the endospore, you can basically control all of these that are underneath it on this list of susceptibility ratings. And so, of course, now we understand prions are even harder to control because they're more resistant to destruction. So when we talk about this relative susceptibility scale, you can see at the top we say they're most resistant. You can think of those as being the toughest, right? But you might also see them described as least susceptible. So don't just look at the word most and least. You've got to use the combination of the words, right? Most resistant is the same thing as saying least susceptible. It basically means they're tough, they're hard to kill. On the other end of the spectrum, we have the most susceptible or the least resistant. So again, these would be the wimpy ones, the ones that are easier to destroy. So if we look at the characteristics of each of these organisms, what puts them in the place on this scale is what I want you to focus on. So you won't have to repeat this scale in its entirety, but you should understand the characteristics about each of these groups that puts them in the place on this scale. So for example, we know prions sit at the top of this scale. They're toughest because they are protease resistant. All proteins should be able to be broken down by proteases, but they are not. Endospores, recall that we built that very thick, very sturdy spore coat that had the dipiclinic acid and calcium that's nearly impossible to penetrate. So that is why they're up at the top of the scale here, or near the top of the scale. Recall that mycobacteria, such as tuberculosis and leprosy, they have that waxy cell wall. That makes it hard to destroy because that wax keeps anything that's water soluble doesn't want to penetrate that cell wall. So these guys are tough to deal with because of that. Protozoa, remember when conditions get harsh, they can actually hide out in a cyst. They will encyst, as we call that. And so it's almost, as tough as an endospore. It's hardy, but it's not quite as tough as bacterial endospores. Even active stage protozoa, even though they don't even have a cell wall to protect them, the fact that when they are in their active trophozoite stage, that they can literally swim away, right? So they're hard to deal with in water bodies and things like that because when they're active, they can just move away from the thing that is harmful to them. Look where gram-negative bacteria sit in comparison to gram-positive. Now remember, even though gram-negative bacteria have a very thin cell wall, that outer membrane, that third layer of protection, actually puts them higher on the scale than even fungi. So that outer membrane is very protective for them. The cell wall components of fungi put them even below that gram-negative bacteria. It's harder to kill E. coli than it is a yeast. So those cell wall components are protective. That's what puts them on this part of the scale. Non-enveloped viruses, we just talked about that. If that's your only covering, is your capsid, then it's going to be very thick, very sturdy, and hard to destroy. Gram-positive bacteria, it's that thick cell wall that makes them a little bit better protected from even enveloped viruses. So they're not at the bottom of the scale, but they're pretty close, even with that very thick cell wall. Enveloped viruses, of course, they're pretty much the wimpiest thing out there because that envelope is made of a phospholipid envelope that they didn't even make, right? They can't control it, they can't repair it, they stole it. And this is why things just like alcohol, hand sanitizers that have alcohol in them, it can disrupt that envelope virus, and that's why they're effective during cold and flu season, because they can damage those flu viruses quite readily. So again, you kind of just, from the perspective of what is it that we need to control? Well, we don't know what's in the environment that we're trying to control. So if we aim to the top of this and use autoclaving to damage endospores, then by default, anything else on this scale that's there, that's present, should be destroyed in the process. So let's run through some types of methods. So we're going to have physical methods and we're going to have chemical methods. When we talk about physical methods, heat is one of the most common ones. So when we say moist heat, right, if you have moist heat plus pressure, that's autoclaving. It's essentially pressure cooking, right? Dry heat would be, for example, the oven. Now, obviously, moist heat, like boiling, is much more effective than dry heat. You could put things in the oven to sterilize them, but without the water to transfer that heat into the substance or the surface that you're trying to clean, that water transfers heat quite readily. So you can always think about, you could put your hand in an oven that's set at 350 degrees for at least a few seconds without burning, but you couldn't stick your finger in boiling water, which is only at about 100 degrees, and not damage the tissues, right? That moisture really transfers the heat quite readily. The pressurized heat would include things like pasteurization. And again, those are methods that we would use that are physical. Cold depends on whether you're trying to just inhibit or kill. Refrigeration is very good for cold, for inhibiting bacterial growth. In fact, that's how we deal with our cultures in the microbiology lab over the course of a semester. You actually can keep them alive for several months if you refrigerate them in between cultures. But if you freeze organisms, it depends on whether you flash freeze or slow freeze. If you flash freeze bacteria, you can actually keep them alive. You would just have to rehydrate them on the other end. Why slow freezing would actually kill a living thing is because ice will form, and that ice will puncture the cell membrane as those ice crystals form slowly. In fact, that is one of the requirements for things that, in this country at least, when you eat raw sushi, that fish, in order to be used, proper food handling laws in this country say that fish had to have been frozen for at least 48 hours. And that's so that if there's any larvae, if there's any eggs or larvae embedded in that fish, if it was an infected animal when they harvested that fish, then it will kill those larval stages in the freezer before you go and serve it raw. Now, that doesn't necessarily mean that everywhere you get raw fish that they've done that, but that is the law in this part of the world. What we call lyophilization. This is actually freezing under vacuum. So it's kind of like freeze drying. But again, that can control the organisms, but it will only inhibit them because that's actually how we buy the bacteria that we use in our lab, because they can be stored for longer periods of time that way. Desiccation, of course, if you just dry something out, all living things require water for metabolism. So if you dry them out, then you have controlled the microbial growth. Filtration, we can filter air, we can filter fluids. And so when you think of things like the N95 mask, that is a filtration form that we use when you enter a room where a patient with tuberculosis is, for example. That face mask would protect you from breathing in any of those bacteria. We would also probably use it in the case of a situation where you have a patient with really suppressed immune system, and you're basically filtering what you're breathing out so that they are not exposed. So filtration might be used on the air in a hospital room, might be used on water, for example. We can think of physical filters, how we physically filter water. or even the culture media, right? How we end up with sterile culture broth is actually they run it through a filter. So it really depends on just the size of the openings in that filter, how effective it is. Osmotic pressure, so if you expose an organism to high levels of salt, for example, it's going to draw all the water out of those cells and keep them from growing. So we talked about halophiles, those that require some salt, but usually salt is very inhibitive. Radiation, things like ionizing and non-ionizing, we'll look at an example of that, but that's UV or X-rays. Those are used sometimes in the food industry now. You might see things like strawberries. They might run them under an ionizing radiation filter, so to speak, and it would kill any bacteria on the surface of that strawberry and keep it from degrading so quickly. So it's a way of increasing shelf life. And then finally, what we call degerming is really just the physical scrubbing, right? When you use a scrub brush on your fingers, that is the degerming stage. If you use a compound, then that would be something else. But the degerming stage is actually the physical process of moving things off the surface of either your tissues or some substance that your equipment that you're scrubbing clean. So your book has a nice diagram that kind of puts all this together. So there's your two forms of heat. Incineration, of course, is a form of dry heat. That is the ultimate form of sterilization. It is the only way to destroy prions, but clearly not everything in the healthcare system, and clearly not human tissues. We can't incinerate everything. It would be, that is how we incinerate biological waste, which is somewhat unfortunate because that is where we put off really toxic compounds. When we incinerate plastics, which is used heavily in the medical field, that puts off some pretty toxic substances. So it's really unfortunate that incineration is the standard how we deal with medical waste. If you look over here under the moist heat, we have things like autoclaving would be steam under pressure. That's what's required to get that moist heat past the endospore to actually kill that endospore stage bacteria. If you boil water or even pasteurization, it's not good enough to sterilize. It's really just disinfecting that substance. You could sterilize something using boiling water, but it would have to be boiling for more than 10 hours to actually get past that endospore. So clearly that's not feasible in most cases. So for example, boiling water used to clean baby bottles and nipples and things like that associated with that, they're not actually sterile, they're just disinfected. Radiation, again, things like ionizing, non-ionizing. Non-ionizing is UV radiation. It's not capable of killing everything, but it's used, for example, you might have seen on toothbrush head covers, right? They have a little UV light filter in there, and it can disinfect the surface of that toothbrush. So it is effective in the sense that you're not trying to penetrate the materials that toothbrush is made of. If you just get the surface cleaned, then it's probably more effective. But ionizing radiation would be things like X-ray. It's actually going to penetrate the tissues. Now, when we say X-ray using to sterilize, clearly it's a very low level energy. You would have to use really long exposures to sterilize certain things because they don't penetrate very well, like for metals and things like that. But these are your physical methods of control. Mechanical methods of control, really sort of a subtype of physical. We talked about filtration, right? So if you have small enough openings in that filter, you can filter either air or liquids typically, but it's really hard to remove everything unless the filter has a small enough filtration rate. So this table came from the old textbook, but I like how it puts it together and kind of wraps up and summarizes some of these things. It even gives you a little bit of detail of what they mean by autoclaving, what conditions actually are met when you are autoclaving. You have to reach a temperature of 121 degrees Celsius at 15 minutes. Now, technically we actually know it could be done in 10 minutes, but there's always a little window of a safety window that's built into those processes. If you look at pasteurization, there are things now on the market that they call flash pasteurization or ultra pasteurized. And these, instead of going 30 minutes, they can go, very quickly. 15 seconds at 72 degrees is enough to pasteurize certain things like fruit juices and things like that. If you were to go to that high temperature on a dairy product, it would actually denature it and it would cause it to curdle, and it wouldn't be the consistency that the consumer wants. So it really depends on what it is that you're trying to pasteurize. Hot air, incineration, we mentioned down here, this basically oxidizes everything it comes in contact with, which is why we can use an incinerator to sterilize the loops that we use in a microbiology lab. Those inoculation loops are usually made of a special kind of metal that can tolerate being heated and cooled to very high temperatures. They're usually very sensitive to physical pressure, meaning if it's in that red hot stage at the sterilization temperature of about 1500 degrees, it would actually break quite readily if you were to put any pressure on it. So it's really specialized metals that can tolerate that condition. We talked about freezing, refrigeration, desiccation. Those things can help inhibit the metabolism of the organism. So again, it's not going to kill everything necessarily, but it can at least preserve food better, and buy some time to control those organisms. We mentioned lyophilization is freeze drying, filtration, we talked about osmotic pressure. And again, this is historically before we had refrigeration, we really were using osmotic pressure, right? By salting the meat, salting the pork. If you use really high concentrations of sugar when you make jelly, that's essentially pulling the water out of any bacteria that are present, and that's going to preserve it. So it's not just for taste, it's actually a control method as well. So we can also use chemicals to control microorganisms, but this really varies depending on the chemicals. Some chemicals are very sensitive to temperature changes and pH changes. Chemicals are only really effective at a certain concentration. They can maybe become less stable over time, so we need to consider how fresh they are. But most importantly, chemicals can only do their job if they've come in contact with the surface that you're trying to control for long enough, right? There's a sort of a saying in toxicology that the dose makes the poison, but the exposure has a huge part of that as well. If you're not, if that substance isn't in contact with the surface long enough to do the job, then it won't be very effective. One reason why chemical methods have to take into consideration organic matter, so think bodily fluids or feces, blood, those things, if they're on the surface of the equipment that you're trying to disinfect, they might actually interfere with those chemicals. The chemicals might not be able to penetrate those compounds and get to the surface and destroy any bacteria or whatever's on the surface. So you have to consider how much organic matter is present under certain circumstances. Most chemical agents are going to require some sort of a solvent, and so if it's aqueous, it's going to be using water as a solvent. If it's called a tincture, things like betadine, right? Betadine is a tincture of iodine in alcohol. And so many disinfectants and antisepsis use tinctures because things that are not water soluble need to be dissolved in a solvent like alcohol. So some example of these chemical methods, we're talking about phenols. You may recall we talked about phenols as being something that they used way back when to prevent surgical wound infections. And so this includes the family of phenolics as well as just true phenols. This is like clove oils or pine sol. Alcohols, of course, we're going to talk about halogens such as your iodine or chlorine in bleach. Oxidizing agents like hydrogen peroxide. Surfactants are essentially things like soaps and detergents, right? When something is a surfactant, it reduces the surface tension And so this is the reason why washing your hands with soap is much more effective, right? The washing of the hands, the physical rubbing of the surface is the degerming part. But if you add a surfactant to that, you will make those bacteria more likely to be removed from the surface of the skin and go with the water as it's rinsed away. And so one reason why we really encourage hand washing, because we're talking about all these methods that are good at killing things, but not all methods are good at killing all organisms. And so when you do degerming with a surfactant, you're not prioritizing any particular organism. You're just physically moving it off the surface of your body. And so it doesn't matter how resistant it is, if it's antibiotic resistant or just something like an endospore. If you physically move it off your skin, then it's not going to be able to expose you, or it's going to be, it's going to go down the drain, rather be someplace where someone else can be exposed to it. So this is one reason just the basic concept of hand washing is so important for people to understand. Using a hand sanitizer that's just got alcohol in it isn't going to kill everything. So that's never a good substitute for washing your hands. Heavy metals, we talked a little bit about syphilis being treated with heavy metals, arsenic and mercury and things like that. But we also use heavy metals to treat things like water. For example, think way back, the Egyptians used to keep water in copper vessels, copper pots, because they figured out that the algae and fungi wouldn't grow quite readily in those circumstances. If it was in a copper, which is a heavy metal container, then it would be preserved. And that's why, because heavy metals actually inhibit bacterial and microbial growth, I should say. Aldehydes, of course, probably the first thing that comes to mind is formaldehyde, but we also have glutaraldehyde, which is one of those compounds they use, for example, in a dental office, that fluid that they drop, like the dental mirrors or something that's not actually been used invasively, they can drop it in that glutaraldehyde and it can be disinfected quite readily, quite quickly in that kind of substance. Gaseous agents, we use these quite extensively in medicine now. What substances or equipment that can't go into an autoclave might go into a gas sterilization chamber. And so when you get back packages that have been sterilized, sometimes they have a little tape or something across the packaging that says sterile. A lot of times in medicine today, it'll actually say gas sterilized. And so it's just a different form of sterilization. Enzymes, we talked about things like pre-enzyme. That's really the category. So again, here in your book, you have this sort of summary table that puts together these. So think about when it's something that's in a chemical form, it can be a gas, a liquid, or a solid, really. But those gases, depending on which form it is, you can use gas sterilization, or some can be used a little bit less stringently to just disinfect. Liquids like Pine Sol and things like that, depends on what you're trying to do and what kind of substance you're using, what level of control that you're going to achieve with those. And of course, you'd have to choose those wisely, because certain liquids can't go on tissue, so you would only be able to do antisepsis with certain compounds. So again, here's a nice summary table. Phenols, again, things like Lysol and substances like that. Actually, those are quite strong odors, and so because they can be sort of irritating, they came up with a slightly different family, what they call the phenolics, and these are very similar. They're just usually a little less toxic in that regard. Alcohols, we said, can really only disrupt membranes, and so they're good for things that are going to be antiseptics, and they can be used as the solvents in tinctures, but they can't kill everything. So by no means do you need to memorize exactly what action, is it targeting proteins or is it targeting membranes? This is a nice summary table, but I'm not going to ask you to that level of detail. Understand in general that chemicals or physical methods can either disrupt their outer coverings or go after proteins or nucleic acids, but you don't have to memorize exactly which one does. Each one does. If you can think of when you approach these different methods of control we're talking about, if you can approach them from the aspect of what are the advantages and disadvantages, so for example, alcohols, the advantage and the disadvantage really both have the same answer. The advantage of alcohol, one of the biggest advantages, is that it has a low evaporation point, meaning it's not going to stay on the skin for very long. So in that regard, it's an advantage because it doesn't leave behind a residue. But the fact that it does evaporate quickly is also sort of a disadvantage because it may not be there long enough the exposure length might be too short to actually kill everything that it comes in contact with. So when you're thinking about any of these, sometimes the disadvantage is just the fact that it's toxic. If the advantages would be, what circumstance would that be the best choice? You could think of that from the advantages point aspect. And then for each chemical or physical method, be sure that you can at least give an example of those. So, or be familiar with the common examples, right? So bleach, for example, is a chlorine containing substance. That would be a halogen. Detergent, right? That would be a surfactant. Copper, arsenic, those are heavy metals. So at least be able to put those examples in the right type of method, and then be familiar with where those would be the best choice, so to speak. And then at the bottom of this chart, you see antimicrobials. That's again a very broad word. That's typically what we use to describe antibiotics. When we use the word antibiotic, we're really talking about the original natural substance that we've extracted from a living thing. If it's called a semi-synthetic or a synthetic, we're talking about antibiotics that we made in the lab based on those natural substances. So they're really kind of all lumped together as antimicrobials. But we'll talk about those in chapter 10.